Dr. Michael Chapman is a research cell biologist at the Alliegro Laboratory at Marine Biological Laboratory in Woods Hole, Massachusetts. He's an alum of the botany program here at UMass Amherst. He has co-authored numerous research articles with Lynn, and I'd like to introduce Michael Chapman.
How is this? Can you hear me? There's a certain amount of overlap between Professor Lascano's talk and mine. That's okay because I plan to be concise. I'd like to tell you a little bit about some collaborative work that Lynn and I did together over a period of 3 years leading up to her passing away. And this work produced the book Kingdoms and Domains, which is on sale in the lobby, and which is a reference volume, a one-volume encyclopedia, if you will, with 3 to 4 pages describing each and every phylum of life on Earth. And it's organized into the 5 kingdoms of which Professor Lascano spoke. And Lynn liked to describe our work on this volume as work on the Linnaean task. So here we have Lynn, and here we have Linnaeus. And the Linnaean task is not just cataloging, but also organizing the different taxa, the different forms of life on Earth. And Lynn approached this task, as she did all of her work, with delight, because for Lynn, work was play. Okay, as Professor Lascano mentioned, the most widely accepted scheme of organizing life is the phylogeny published by Carl Woese and colleagues in 1990, which is it's called the 3 domains of life, or the tree of life. And the 3 domains are shown here: bacteria, archaea, and eukaryotes, and this tree is based on comparative sequences of ribosomal RNA. Now, the ribosomes are the factories of the cell. They read the messenger RNA and they produce the proteins, and as such, they're extremely essential components of the cell, and they evolve at slow rates. Their molecular clock, as it were, is very slow, and that makes them suitable, their sequences suitable for parsing evolutionary relationships between big groups, between phyla of life. And one can produce a tree like this, wherein the lengths of the different branches reflect degrees of relationship, degrees of relatedness between different taxa. But one also notices when one looks at this tree that it's all bifurcating. There are no what Lynn called anastomoses. The branches diverge and diverge and diverge again, but at no point do they come together. So in other words, this tree leaves no conceptual room for any sort of symbiogenesis, for any sort of hybridization, for any sort of lateral gene transfer. And Lynn felt that there were other aspects, other differences between the organisms of life on Earth that were not reflected in this tree and that were very important. And they're related to this event here, the origin of the first nucleated cells, which took place approximately 2.5 thousand million years ago, and which involved also the acquisition of chloroplasts and mitochondria via symbiogenesis, all under selective pressure of the oxygen atmosphere. Now, Lynn's argument, which I found very persuasive, was that archaea and bacteria had much more in common with one another than either group had with eukaryotes. They have no membrane-bounded organelles. They have no nucleus. They have no cytoskeleton. They are unicells, order of magnitude smaller than eukaryotic cells, whose DNA is organized in a circular fashion. And all these are valid aspects of the bacteria and archaea, which should be taken into account in cataloging them, in organizing them. The nucleus is an incredibly complex entity. Its chromatin is organized in linear DNA complexes with protein as opposed to circular. It's bounded by a double membrane with pores for the transit of messenger RNA or proteins. And it's dependent on the intracellular motility system, which forms the spindle for mitosis. Without the spindle, the nucleus would be not much good at all. This system of the nucleus and the cytoskeleton, the nucleus and the central kinetosomes, is totally interdependent. And suggests that it came in as a package deal. So Lynn, in order to express these important points that I just mentioned, where did the nucleus come from, the mitochondria, the chloroplast, she came up with a 5-kingdom network of life, which shows these anastomoses, which are at the gray triangles. The lowest one, where bacteria and archaea came together, we know from protein studies that the eukaryotic cytoplasm contains proteins from both lineages. Then the middle one represents acquisition of mitochondria, and the top gray triangle, acquisition of the chloroplast. But in other words, what you have here in the 5-kingdom scheme is not a tree or a bifurcating network, but rather it's a net. And in terms of accuracy, I liked what Professor Lascano said about which one of these you choose depends how you look at life, because they're both completely valid. One might also consider the means of cataloging books and arranging books in the medieval library, when books were so valuable that they were still chained down. At first, they were ordered in order of acquisition. Now, one can imagine how helpful that would be as soon as you get past 20 books or so. But yet, it's a valid scheme of organization. So when Lynn said that the 5-kingdom scheme was more teachable, I've said it's more accessible. I would choose this one every time, especially when dealing with beginning students. Now I'd like to talk a little bit about this organelle here, which was very important to Lynn, and she insisted that it be called an unulopodium. These two cells are not drawn to scale. The prokaryote on the left should be like 100 times smaller than the eukaryote on the right. It would appear to be a speck on the cell membrane of the eukaryote. And it has 7 flagella, whereas the eukaryote has 1 unulopodium. Now, what's the difference between these, and why is this important? Well, because the unulopodium contains elements of the cytoskeleton that I'll show you in more detail in a minute. And this cytoskeleton, the nucleus is completely dependent on, and it also depends on the nucleus, which encodes the genes. And this collective system, the nucleus plus cytoskeleton, plus the nuclear connector, and plus also, it turns out, Golgi apparatus, is referred to as the karyomastigont. To give you a little more detail, here's a bacterial cell with its flagellum. The flagellum runs by chemiosmotic mechanism of proton flux through the cell membrane, which drives a rotary motor. Now, this rotary motor has been presented by Michael Behe and others as indicative of intelligent design. It's such an efficient motor. It's so complex that it must have evolved as a unit. It must actually have been created as a unit. And so he says intelligent design. Now when I meet someone who doesn't believe in evolution or who challenges my belief in evolution, my use of evolution in my everyday work, I say, if you feel that way, why not save some money and take last year's flu shot? So here are these two organelles in cross-section. The flagellum is composed of just one protein. It's called flagellin, and it traverses the cell membrane. The underlipodium has at least 360 different proteins. These proteins comprise the microtubules of the axoneme that you see in their 9 pairs and 1 central pair here. It also involves the radial spoke proteins and the things that look like little feet that are coming off one member of each pair of microtubules. Those are ciliary dynein. They're specialized motor proteins which in the cytoplasm, they walk along microtubules as though the microtubules were highways in the cell, and they move things like organelles around the cell as needed. In the case of the ciliary dynein, though, they're fixed to the microtubule pairs, and there are docking sites on the adjacent pairs, and they cause one pair to slide relative to the next one, and that's the mechanism whereby the underlipodium can whip. Okay, and this the flagellum is 16 nanometers across. Just one of these microtubules is 24 nanometers across. The underlipodium is bounded by a membrane instead of traversing the membrane. Other differences abound. I'm showing you this because I want to emphasize how universal this structure has been in evolution, and how the underlipodium has actually can now actually be found in the retinal rod cells of our eyes, in olfactory receptors, mechanoreceptors, audio receptors in the vestibular system. They help us maintain our balance. And they are remarkably standardized throughout the protoctists, plants, and animals. So in other words, there are not just hundreds, but thousands of taxa which have this same exact, very complex structure with the 9 pairs, the central pair, the motor proteins, the nexins, and what have you. So what this suggests to us, to me, to Lynn, to our colleagues who also are interested in studying symbiogenesis, is that this organelle came in by lateral gene transfer, either by symbiogenesis or by acquisition of genes through some other mechanism, viral transduction, if you will. But that at least the underlipodium and maybe the entire karyomastodon is the product of symbiogenesis or some kind of lateral gene transfer and originated as a unit. Now, Lynn spent maybe the last 20 years of her life trying to establish whether or not spirochetes contributed to the origin of the underlipodium. There are spirochetes which are roughly the same size and shape as an underlipodium. One can convince oneself that they swim with a similar waveform. To me, it's never looked exactly the same. But one thing's for sure, that free-living spirochetes, many of them are poisoned by oxygen. And so this oxygen selective pressure that I mentioned earlier would have behooved these guys to find some sort of refugium, some way to get away from the oxygen. And one way to do that would have been to associate themselves with something like thermoplasma, which uses sulfur as its terminal electron acceptor in much the same way we breathe oxygen, and produces H2S gas. And this H2S is a reducing agent which would have provided the spirochete with hiding place from oxygen. The spirochete might have become associated with this thermoplasma and conferred motility on it so it could swim off to greener pastures and find more elemental sulfur, if you will. This hydrogen sulfide, by the way, is the source of the marine smell that one finds at the beach. These microbial mats are ubiquitous around the world, and when one finds a white flocculent material associated with them, chances are that this sort of syntrophy is going on. Now, the problem is that nowhere in any spirochete have microtubules been found. Nowhere in any spirochete have protein homologs to dynein or kinesin or nexin or any of these other 360 flagellar proteins been found. They may superficially resemble Underlipodia, but there's no direct evidence for all Lynn's work that they are the evolutionary progenitors. John Hall has found contributions of the spirochete genome to eukaryotic cells through a reciprocal blast technique, but these proteins had nothing to do with motility. They're proteins of intermediary metabolism. So the question remains open. The question remains open exactly how the karyomastodon evolved. There's still some question whether it's actually primitive, basal, I should say, to the eukaryotes, although it's found in things like termite hindgut protists, anaerobic protists, which are considered to be basal eukaryotes. But we explore these questions in text boxes throughout our book, Kingdoms and Domains. We have boxes on hybridization and polyploidy, boxes on mitosomes and hydrogenosomes, which are organelles related to mitochondria, and also on modes of multicellularity, how multicellular condition evolved from unicells. And so I would encourage you guys to take a look at Kingdoms and Domains on your way out. This took 3 years for Lynn and I to produce, and we dealt with Elsevier Academic Press, which was not unlike dealing with the Soprano crime family. But in any case, here it is. And here's Lynn, the way I remember her best. I had no time to hate, because the grave would hinder me, and life was not so ample I could finish enmity. Nor had I time to love, but since some industry must be, the little toil of love I thought was large enough for me. Thank you.